Hi Jobbies. Welcome to lesson 2 of the cell cycle. So in this lesson we're going to discuss mitosis. So like I said, the cell cycle, I divided it into 3 lessons. The first one we did the overview. We looked at the cell cycle in general. So now we're going to mitosis. So that will be the main focus of this lesson. And then the last one, which will be meiosis. So just a quick recap on what was discussed last time. We looked at the cell cycle and we looked at interphase. So interphase, while we discussed it, is made up of G1, S, and the G2 phase. So just to have a further recap, so we said this is like the cell. So this is the cell cycle, and then we said we're going to have G1 phase, S phase, and the G2 phase. And then this is the mitotic phase. So this mitotic phase will be the focus of this lesson. So previously we discussed from G1, we looked at the checkpoint, the DNA replication phase, and then the gap 2 phase, the second gap phase. So this is what we called interphase. So from G1 up to G2, that's interphase. And then after G2, we're going to have a mitotic phase. So this interphase is what we looked at. And just an important concept is that this phase is longer than mitotic phase. But in explaining it is even short. So now let's go to today's lesson. So in today's lesson, we're looking at Cytokinesis, and then later on we'll do Cytokinesis. So we did interphase. Now we have come to mitotic phase, where we're going to look at Cytokinesis, which I said it's nuclear division, and Cytokinesis is cytoplasmic division. So now in discussing mitosis, we can't jumpstart to go to mitosis without first having a quick discussion on chromosomes. So we should first talk about chromosomes, and that's when we'll go to mitosis. So let's discuss chromosomes. So that's the introductory part of this lesson. So what are chromosomes? Chromosomes are thread-like structures that carry the genes. So the genes, they are simply DNA segments. So now how does a chromosome actually look like? So a chromosome will look like this one here. So this is a chromosome. It's thread-like. It's like a thread. So a chromosome is made up of 2 parts. We have one chromatide and another chromatide here. So this is a chromosome. So a clear diagram is this one. Under a microscope, you should be able to see this. So these two, they are called sister chromatides. And then now we should understand from ground level, how does a chromosome come about. So I'll take you back to what we discussed earlier. So we discussed that DNA is a double helix, where we said we have adenine, we know it pairs with thymine, and then guanine will pair with cytosine. Again, adenine pairs with thymine. So that was this is more of a revision of last time's work. That was semester 1. Okay, so the double helix comes in because of this. So we're going to have one template and another template there. So the DNA double helix will look like this. So this is just for explanation. Yeah. So this is the DNA double helix. So this is under, it can look like this under an electronic microscope. Okay. So this is the pairing. So these they are very microscopic. So the DNA double helix can actually look like this. Yeah, and then when you magnify it, that's when you'll be able to see this. Yeah, so we're starting with this so that we have a detailed understanding of chromosomes. So DNA is this thing. This is a gene. Yeah, so we're going to have a gene, which you know a gene is a basic unit of inheritance. A gene is the basic unit of DNA. So now you know DNA is made up. We say DNA is protein in nature. So what does that mean? When we say DNA is protein in nature, it's because of a structure called the histone. So we have this structure which is a histone. So the histone, we can think of it as a bead. So this histone, it is the one that is made up of protein. So we have the histone here. So what we're going to have is whereby this histone, so the histone is there for stability, to make the chromosome more stable and the like. And the DNA, which is the gene, this one, it carries genetic information. So what happens is that the gene will wrap around the histone. Yeah. So the gene, these genes, they'll be around the histone. So in any form, so it's just around the histone. So the complex by which the gene is around the histone, where the gene wraps around the histone, we call it a DNA histone complex. So, or you can, other texts will say gene histone complex. Yeah, so this will now form a more stable structure. Okay, so I'm just moving in layers. So now this structure, which is made up of a gene and a histone, it is called a nucleosome. So this is called a nucleosome. A nucleosome is made up of a gene, or you can say DNA, together with a histone. So this is why we say DNA is protein in nature. Yeah, so you can answer something on an essay question where they say explain. Yeah, you can research even more. So this is the overview in summary of the whole lot of things. So now this nucleosome, the nucleosomes will now be a lot. So like we're going to have another nucleosome. So we're going to have another nucleosome. So we have another nucleosome, and then it will also have genes strapped around it. Again, we would have another histone. Oh, so this is a histone with DNA, meaning it's a nucleosome. And then we're going to have another histone again with DNA wrapped. So now, if we have a lot of these, if we're going to have a lot of nucleosomes, we would have something like this. So this would be like nucleosomes. So nucleosomes will have they'll be wrapped by DNA. Yeah, so the more nucleosomes we have now, when we have a lot of them together, they are going to form what would be called a chromatide. So a chromatide. Okay. So okay, so I hope you have seen where we're going. Yeah, so we're just having the development of the chromosome. Yeah, so you have now known that this is a chromatide. A chromatide is made up of nucleosomes. Nucleosomes are made up of histones, and histones are wrapped around by genes. So these chromatides now, when we have two of them, when we have two chromatides together, these two, they'll be what we call a chromosome. Okay, so together these two will form a chromosome. So I've helped you understand how these they flow. So it can come in form of a multiple choice. So you can be asked something like it can be which one is the correct order, and then you are given multiple choice. You are told the first one, they give you a gene, and then they give you a chromatide. They give you a chromatide, and then next you are given a chromosome, and next you are given a chromosome, and then the last thing you get a nucleosome. So after chromosome, you get a nucleosome. You pardon this laziness. So you have multiple choices. They'll swap these. So for you have to identify the correct order. You should be able to say, okay, so the smallest unit is a gene, and then after a gene we're going to have a histone. Yeah, so there'll be different options. Yeah, so I haven't managed to find the past paper which has these kind of questions. So after a gene we have a histone, and then after a histone there can be DNA histone complex. So this gene and the histone together they will give a nucleosome. They'll give a nucleosome, and then after a nucleosome, that's when we have a chromatide. And then two chromatides will give a chromosome. Yeah, I'm sure you've understood very well. Yeah, so from here now, we're now going to look at before we go to mitosis, let's also discuss a little bit further on the chromosome structure, what it has, and the important structures that will guide us now to mitosis. And before that, let me just quickly show you the arrangement of these. Okay, so just for you to appreciate more, so this is the DNA double helix, so this is why I was saying the DNA is looking like this. Yeah, so this is just a diagrammatic for me to explain to you. Okay, so this is the DNA. So the DNA, which are the genes, together they'll go and wrap on the histones. So these two are beads, the two are balls, these which you can see here. Yeah, so should I zoom in? Okay, so I hope you're able to see. So these, the histones, they'll be wrapped around by the genes. So we have another histone here and a gene. So together they form a nucleosome. Yeah, so we have a nucleosome, 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 nucleosome. So the whole lot of these nucleosomes together, now they'll start curling up, and then in the end they'll form a chromatide. So these nucleosomes, they'll form a chromatide, this chromatide here, and then two chromatides, they'll make up a DNA. Okay, so a chromosome rather, sorry. So we've discussed a lot now on this. So let me quickly now rush you on the chromosome structure. So the chromosome, for explanation purposes, we're writing it like this, like an X. So the important structures on the chromosome, which you should know, firstly, is we have these sister chromatides. They are sisters because they are found together. They are made up of the same genetic material. So we have these two sister chromatides. And then from two sister chromatides, we will now have the middle part here. This is the centromere. So the middle part is called a centromere. So we have a centromere. Yeah, so I won't write it in full. I'll show you the diagram. Okay, so we have a centromere here, and then what else do we find? So at the center here, the centromere, we have cohesion proteins. So at the center here we have cohesion proteins. So the cohesion proteins, their function is to bind these to the sister chromatides. So we have these two sister chromatides. So at the centromere, at the center here, there are cohesion molecules which bind. Yeah, so sometimes not necessarily living at the center, they can be distributed around, so they bind the sister chromatides. Yeah, so the cohesion proteins bind the sister chromatides, so take note of that. And then the other very important structure is the kinetochore. So we have kinetochore. Okay, so we have kinetochores. So this is C. Kinetochores. So the kinetochores, they are found here again. So they are disc shaped. So you can imagine them as being something like a disc, the kinetochores. So these kinetochores, they are very important because this is where there will be attachment of microtubule filaments. So we haven't even started discussing, and we're talking about a lot of things. Okay, so we have microtubules. So we have, so this is just like a simple chromosome. So on the chromosome here we have kinetochores, and then these kinetochores have attachments which are called microtubule kinetochores. So we have attachments like this. So here we're going to have attachments to the kinetochores. So these, they'll be very important later on when we now go to anaphase. Yeah, so we have kinetochores. So this is the basic thing you should know about the chromosome. Yeah, we've talked a lot now. Let's get down to business. So before that, let me show you this. Okay, so this is a beta structure of the chromosome. Yeah, so we've discussed the chromosome. You know how it comes about: gene, histone, nucleosome, chromatide, and the chromosome now. Yeah, so we have this chromosome. Okay, yeah, so you can see we have a chromatide here and another chromatide. At the center we have the centromere. So this is the centromere, and then the same. Okay, and the structures I wanted to explain very much on these. So this yellow thing is the kinetical. Oh, it's here. So this is the kinetical, and then the kinetoco, it has the microtubules. These, they are the ones which would separate the chromatides. So you'll see the chromatides will be separated, they'll be pulled. Yeah, and then take note also that the attraction between the two sister chromatides is as a result of the proteins. What did we call those proteins? The cohesion proteins. Yeah, so now we've now discussed the chromosome way more than I planned. Yeah, so we're done with the chromosome. Now let's go to mitosis, the main deal. Okay, so we can go to mitosis now. So what is mitosis? Mitosis is a type of cell division in which one cell, the mother cell, okay, so in which one cell divides to produce two cells. Yeah, so we're going to have the mother cell producing two daughter cells. Yeah, and then those daughter cells will be genetically identical. Yeah, so this is the whole thing on mitosis. So to sum it up in form of a diagram, we're going to say mitosis is whereby we have a mother cell, and then in the end the mother cell will produce two daughter cells. So the DNA which will be in this mother cell will be the same DNA which will be in these two daughter cells. Okay, so now we're looking at the cell cycle. So now this is now the mitotic phase. So meaning that we're going to have different phases in which mitosis takes place. And previously we discussed we talked about the mnemonic which can help you to remember. So we said the phases of mitosis can be remembered by the mnemonic Professor Matt. Yeah, so we have this Professor Matthews. Okay, so Professor Matthews. So these, they represent the stages. So the first one, it is prophase. So the first mitotic phase is prophase, and then the second one, M, it's for metaphase. Okay, so the second one, it's metaphase, and then blah blah blah. The third one, it's anaphase. So I have anaphase there, and then the fourth one will be telophase. So we have telophase. Okay, so the And then one addition now, dealing with mitotic phase. So on the first one we have prophase. So prophase, this one, after prophase, there's a phase which happens in between prophase and metaphase. So that phase, it's Prometaphase. So we have Prometaphase. So this is the phase which takes place between here. After prophase, we have Prometaphase, and then that's when we go to metaphase. So this is Prometaphase. Okay. And then so now from here we'll now go straight to discussing each one of these. Yeah, so I'll make sure to be brief on these. So let's start with prophase. So we'll look at the condition of a cell at prophase, and then we go to prometaphase, just like that, proceeding. Okay, so getting started with prophase. So now firstly let's draw the cell. So this is the cell and it has a nucleus. So usually we just shade the nucleus. So in the nucleus, that's where we find DNA. And when we're talking about DNA, you get that we're simply talking about chromosomes. Okay, so now what happens under prophase? So the first thing that happens here is the condensation of the chromosomes. Yeah, the DNA condenses. So what does this mean? So this is this cell, and then we have this nucleus. So initially we don't see the chromosomes. Yeah, but under prophase now, that is where condensation takes place. So the chromosomes will now start appearing. You'll be able to see the chromosomes. You see the chromosomes. So for this explanation, we'll just use up to like 4. But you should be able to understand that there are 23 pairs of chromosomes. So like this is a pair, there's another pair, so all the way up to 23. Oh, and I even forgot to mention this when we were discussing the chromosomes. So you can just take note on the types of chromosomes. Yeah, so you can just take note that we have autosomes. We have this is auto, so we have autosomes. We have autosomes and 6 chromosomes. Okay, so autosomes and 6 chromosomes. So these are the major types of chromosomes. So one thing just to know here is that the autosomes, starting with the 6 chromosomes. So the 6 chromosomes, we only have 2 pairs. Should we? Let's say 1. So we have one pair of 6 chromosomes, and then they are made up of 2. Yeah, so for the 6 chromosomes, you know we have XY for males. So this is 1 pair. There'll only be 1 pair in a cell, or it can be XX. So we have 1 pair of 6 chromosomes, and then we have 22 pairs of autosomes. So in the cell, we have a total of 23 pairs. 23 pairs of chromosomes, and then the total number of chromosomes, since this pair is 23, meaning the total is 64. Yeah. So in pairs, when we divide 46, when we divide 46 by 2, we get 23. So I just wanted to make mention of that. So out of the 23, we have one pair which is the sex chromosomes, and then the rest of the entire pairs, they are called autosomes. Okay. So now we proceed to our prophase. So now here we have said the first thing that happens is condensation. So there will be condensation of the chromosome. So condensation, blah blah blah. So condensation is whereby the chromosomes now start appearing, like you can now see them under the microscope. Yeah, so you get to understand that the nucleus is very small, like you can't see the contents, but later on after condensation, that's when you'll be able to see the chromosome structures. And then the other thing, it is where now we have spindle fibers. So let's start with centrosomes. They are what are called so we have a structure called the centrosome. Yeah, so you can just be careful not to mix this. So there are centrosomes, and we also talked about the centromere. Centromere. So we said the centromere is on the chromosome structure here on the middle part where they are joined. That's the centromere. So now for the centrosomes, so the centrosomes now, they are just a center. So like we have centrosomes in the cell. So now under prophase, this is the stage where centrosomes appear. So there will be centrosome and another centrosome like that. So centrosomes appear here now on the prophase, and then after the centrosomes appear, the other thing which is observed under prophase is this. So we have, this is the nucleus. So under the structure of the nucleus, it has a membrane. So now this same nuclear membrane, or we can say nuclear envelope, it disintegrates, so it breaks down. So under prophase, this nuclear membrane which was here, it will now break down, so it will be like in form of dotted lines. The nuclear membrane will break down. Yeah, and then the substance inside the nucleolus will also disappear. Okay, so these are the events that take place in under prophase. So let's quickly go through. Firstly, condensation, we're able to see the chromosome structures, and then the centrosomes appear. The centrosomes will appear in the cell, and then from there the nuclear envelope will disintegrate, or it will break down. Okay, and then the nucleolus inside it will disappear. So this is the prophase. So now we're going to the next phase, which is metaphase. So now coming to metaphase. Oh, sorry. So before metaphase, we undergo prometaphase. So this is the part where I mentioned earlier that between prophase and metaphase, there will be prometaphase. So now let's discuss the events that take place in under Prometaphase. So now on Prometaphase, so this is now the cell is it's undergoing mitosis. So now under Prometaphase, the spindle fibers. Oh, I forgot to even mention one thing. That today isn't my day. My mouse is performing. Okay, so here now under Prometaphase, it's continuing from where we ended with Prophase. So there will be migration of those structures. We said those, they are centrosomes. Yeah, so the centrosomes will migrate to now go to the opposite pose. So from there, one will come here, the other one will go there. Okay, so the centrosomes will now come here now. They'll be at opposite pose. And then after that, the chromosomes will undergo further condensation. Yeah, so this is the disintegrated nuclear membrane. Yeah, and then the chromosomes which are inside, they will undergo further condensation. So meaning they'll now be more visible. Yeah, so we can actually draw them bigger. Yeah, so the chromosomes will undergo further condensation, and then after undergoing further condensation, kinetochores will now appear. So remember, we said this is the structure of a chromosome, and then we have kinetochores here. So let me just quickly show you that picture of the kinetochores. So the kinetochores will appear now. This is at Prometaphase. So the kinetochores, these which are in yellow, they will appear. So we have kinetochores there and there. Even here, they will appear on both sides of the chromosomes. So that happens at Prometaphase. Okay. So now when the kinetochores appear, oh, and I just mentioned these, I didn't even talk more about them, the centrosomes. So the centrosomes, their function is to kind of produce mitotic spindle fibers. So there are structures called spindle fibers. So there are spindle fibers which are produced from here, from the centrosome. So the spindle fibers, they are the ones, so this is the centrosome, and even there we can have a centrosome. So the spindle fibers will come from here. So this will actually happen right here at Prophase. So the spindle fibers will be released from the centrosomes so that they will attach at the kinetochore. So under phase, the kinetochore had to appear. So the kinetochores appeared, and then the mitotic spindle fibers, they will now attach to the kinetochores. So even here, the spindle fibers move from the centrosome going to the going to the chromosome. Okay, so these are happening at the Prometaphase. So let's quickly go through. So we said at the Prometaphase, the centrosomes will move to the opposite pose, and then after moving to the opposite pose, the chromosomes undergo further condensation. After undergoing further condensation, the kinetochores will now appear. And then when the kinetochores appear on the chromosomes, the spindle fibers will now attach. The spindle fibers will attach to the to each of the chromosomes, these chromosomes. So there will be that attachment. And then from there now, we go to the next phase, which is metaphase. So we're done with prophase. So I'm going to give you a summary of the whole lot of this for easy revision. So now that was metaphase, that was prometaphase. So now let's discuss metaphase. So what will happen here on metaphase? So this is our cell. So yeah, okay, let's draw the cell. Okay, and then now so now at this point, this is where this here we had chromosomes here. So this, the nuclear membrane disintegrated, and then it just remained as fragments, these to my dotted lines. Yeah, so let's draw our chromosomes. So we have 1, 2, 3, 4. So there are a lot more chromosomes there. So now what happens here is that the nuclear membrane completely disappears now. The tumor fragments which remained, they'll all go. This is now at the metaphase. So at the metaphase now here, this is where, so here we have the centrosomes and the other centrosome. And then from the centrosome we have spindle fibers. We have these spindle fibers. So there are more because we have a lot of chromosomes. So we have all those spindle fibers. So the most important thing now which you just have to know on the metaphase is that this is where chromosomes are aligned at the equator, at the metaphase plate. Yeah, so this equator, so let me just use a different ink. So here at the equator, this is where the chromosomes will line up. So what do I mean? We're going to have, this is our cell, and then we have the centrosomes here. So these chromosomes, they will now align at the metaphase. They will align at the equator. So this is the biggest event that happens here. And the reason why this happens is because now there is no nuclear membrane. The nuclear membrane here that was putting the things together to be in one place. So the nuclear membrane was disintegrated. So now at this point, these, they are just aligned at the metaphase plate. So these spindle fibers now, they are fully developed at the metaphase. So the mitotic spindle fibers are fully developed, and then these chromosomes, individually, this is a chromatide and the other one is a chromatide. They will align at the equator. Okay. So this is all on the metaphase. I think the metaphase is very easy. So each sister chromatide is attached to a spindle fiber. Okay, and then we're making progress. So remember, it's Professor Matthews. We're done with metaphase. We now go to anaphase. So now what events take place at anaphase? So now this is anaphase. Okay, so here now at anaphase, okay, so here now we continue with the development of this cell. So at the anaphase now, so the most important thing for metaphase is the chromosomes will be at the equator. So now at anaphase, the most important thing is that these chromosomes now, they'll be put to the opposite pose. Yeah. So let us draw so these are our sister chromatids. So we have 1, 2, 3, 4. So these are our sister chromatids. So these chromatids, they'll be pulled now. Yeah, so they'll be pulled to the to the opposite pose. So remember, we have the centrosomes. So the spindle fibers will pull the sister chromatids to the opposite pose. Yeah. So let's do this. So they'll be pulled in. All of these will now be pulled to the opposite pose. So when they are pulled, how will they look? So the structure will now look like so this here we have so like these are the chromosomes. So what will happen is that this one side will be pulled, and then the other side here, it will also be pulled. Yeah, so this is what is happening. They'll be pulling. So one chromatide will be pulled, and then the other chromatide also be pulled to the other direction. Okay, so I made the mnemonic. Now it's very inappropriate. Yeah, so here we're going to have the chromatides being pulled. So we're going to have this is the first one. It's been pulled like that, and then the other one, we'll have the other one, and the other one, like this. So what is happening here is simply the chromatides have been pulled. So I've actually drawn 3. So the chromatides are now being taken to the opposite pose. Yeah, and then we had 4. So all of these, they are being pulled. Yeah, I think that's the major thing you have to know under anaphase. They'll be put to the opposite pose. So now these chromatides, the sister chromatides, so just for demonstration, so we had this. One side has been pulled, it has gone to the other side. Even this, it has been pulled. Okay. So that's on anaphase. So, and what makes them to be pulled is because the cohesion proteins which were holding them together, they will break down. So at this point, the cohesion proteins here, they broke. Even here they disintegrated. That's what happens, and when that happens, then there can be pulling of the chromatides. So these chromatides which were pulled, they will now be called chromosomes on their own. So this was a chromatide, even this was a chromatide, but at anaphase they will now be called chromosomes. They'll individually be called chromosomes. And then the other thing that happens here is that on the interphase, the cell elongates. Yeah, so this is where the cell even elongates more. So like it will be stretched. Yeah. So at interphase, the cell will no longer look like this. It will look something like an egg because there's pulling. So this is on the interphase. And then we go to the to the last phase, which is telophase. Okay, so let's discuss the telophase. So now what happens at telophase? So at the telophase, it is now more like the opposite of anaphase. So this is our cell. So let's draw it like this. This is our cell, and then the chromatids, they have now arrived. We had 4 of them, so maybe they are here now. Even the other ones, we had 1, 2. They are just individually. There are 4 individual chromosomes. Okay, so these, they now reach the opposite pose. This is now at telophase. So under telophase, you just have to rethink anaphase, and then you start making the opposite. Or on prophase. So prophase, we saw the nuclear membrane disintegrated. The nuclear membrane was like this, but it became dotted. Yeah, that was on prophase. So now at this point, after these individual chromosomes arrive at the opposite pose, they will now form a new nuclear membrane. The nuclear envelope, it will now form back. Yeah, so the chromosomes will decondense, and then the nuclear membrane will now form around. So the nuclear membrane forms, and then those tumor centrosomes, they disappear here. Remember, on prophase, that's when the centrosomes appeared, but now at this point they are disappearing. So this is it. This is telophase. So one thing I want to show you is that okay, so we're getting there. So for mitosis, you have to understand that the parent cell will produce two identical daughter cells. So like we're going to discuss more in meiosis. So but here you see that we started with 4 chromosomes. So even when this cell becomes 2 cells, it should have 4 chromosomes. Yeah, but for meiosis, we're going to see that they can be actually 8. So let's go and conclude this side. So this is where we're coming from. We're now here, telophase. So telophase, this is under telophase, the nuclear membrane has formed, and yeah, everything like that. So after telophase, now this is the part where we now come to cytokinesis. So cytokinesis is not part of this cell cycle. So in the cell cycle of gap 1 phase, replication phase, gap 2, and mitotic phase, which we are looking at today. So this cytokinesis happens after the whole lot of mitosis has taken place. So what happens next? So we come to cytokinesis. So now after the chromosomes arrive at the center, there will be formation of a cleavage furrow. So we'll just use the same, this one. So after telophase, now there will be a cleavage furrow. So it is whereby the cell will now, like, it will squeeze in the middle. There will be squeezing. So how does this look? So the cell, it was looking like this, kind of circular, but here there will be now a cleavage furrow. So where now the cell is squeezing like that at the middle here, it's getting squeezed, and then you can see that we have a nucleus here with those new chromosomes. Even here we have a nucleus with those 4 new chromosomes. So the cleavage furrow forms, so that in the end, when it continues, we are going to remain with two new cells. We have this cell and that cell, and then each of these cells, they will now be like the original parent cell. There are 4 chromosomes. So these cells can as well undergo mitosis. They'll undergo prophase, so that again they'll divide further. So this is what happens. A cleavage furrow forms, and then in plant cells, so this is in animals, in plant cells it's a cell plate. This, it's called a cell plate being formed. Okay, so let me show you the summary and then we call it a day. Okay, so this is mitosis in summary. Yeah, so you can screenshot this and use it for your revision for studying. You might be asked to explain all these, so you should be able to draw, you draw and you explaining. Yeah, so we discussed all these. Here the chromosomes becomes visible, the spindle fibers form, the nucleolus, the contents in the nuclear membrane, they disappear. The nuclear membrane, we said it will break down. So when it breaks down, it means here it becomes in dotted form. Yeah, so this was our journey. Prophase, the major thing is that the spindle fibers attached to the kinetochores of the chromosomes. Metaphase, the main thing is that they'll form at the metaphase plate or at the equator. Anaphase, the major thing is that they'll be pulled. And then telophase, the major thing is that the membrane which was broken down, it will form again. Yeah, and then lastly, we're going to have cytokinesis, where there's disintegration now. This will separate. So there's a cleavage furrow or a cell plate. Yeah, so you can use this for your revision. Screenshot this, and while you are not clear, screenshot that part. Okay, so this was our journey with the cell cycle. Yeah, the cell cycle now is complete. So I have another summary for you under mitosis. Here, this was the beginning. So firstly, there was condensation. This was prophase. So remember prophase. Also, this is prophase here, where we're starting from here, and then metaphase, anaphase, telophase. Professor Matthews. Okay, so this was the whole lot of it. Yeah, in the end, we're going to have 2 daughter cells. So according to our objectives, we are supposed to do some practice questions. So that will be in a separate lesson. Yeah, so that will be in a separate lesson. It will be called part 2, lesson 2, part B. Yeah, and then we go to meiosis. So from this, you watch the lesson where we practice a few questions so that we understand these things even better. Okay, so until then, we'll meet for meiosis. For now, it's bye-bye. Continue studying well.